Now, whether I'm audible now? Yes, yeah. Yes. Yes, okay. Okay, any doubt in first session? We have already seen 60 questions. Any doubt is there? If no other doubt means we will continue the remaining 60 questions. Okay? Yes. Be attentive in class. If you have any doubt, kindly let me know. Okay? First question. The unit of leaf area index. First is unit of leaf area index. What will be the unit? Whether it's meter square per gram or gram per meter square, gram per gram or dimensionless. Yes, very good. The unit of leaf area index. First, we have to know the formula of leaf area index. Leaf area index is what? Leaf area by ground area. Okay. If area means what? Maybe like meter square by meter square. So, this may be cancelled. So, it will be a dimensionless. Yes, it will be dimensionless. What is the empirical formula for chlorophyll A molecule? What is the formula for chlorophyll A? What will be the formula for chlorophyll A? C55H72O5N4Mg or C55H70O6N4Mg? What will be the answer? Yes. C55H72O5N4Mg. Okay, this is for chlorophyll A. Why it is hydrogen is 72 means due to presence of the methyl group will be there. Okay, but in chlorophyll B, aldehyde group will be there. Okay, so it will come C55H70O6N4 and Mg. This is for chlorophyll B. I hope this is clear for everyone. Okay, which of the following is not the most commonly used marker gene for transformation? They have given the example of marker genes. They are asking which is not a marker gene among these. Nopolamine synthase, beta glucuronidase, luciferase, yes. Cryonease is not a marker gene, okay. It's a transgene that will used for improving the insect resistance. Okay, insect resistance will be done. For example, bole guard, yield guard, like that we can develop varieties by integration of this cryonease genes. Okay, clear? A polyketide that inhibits the enzyme ATP synthase by blocking the movement of protons through F1 subunit. So, sorry, through F0 subunit is what? What is the inhibitor that will inhibit F0 subunit of ATP synthase? This we have already discussed. Yes, very good. F0, 0 is what? It's a oligomycin. So, is oligomycin will act as an inhibitor for F0 component of ATP synthase. F0 component. Okay. Then another question from plant hormone. Plant hormone bioassay they are asking. The common bioassay used to detect the plant hormone brassinosteroid. In class itself I told. From bioassay they will ask one question. What is the common bioassay used for brassinosteroids? Draft rice lamina inclination bioassay, or cucumber cotyledon expansion bioassay, or triple response in bioassay in etiolated seedling, or oat coleoptile elongation bioassay. What will be the right answer? Yeah, bioassay for brassinosteroids. Triple response, that will be specific for ethylene. Yes, that will be specific for ethylene. Oat coleoptile elongation. Yes, that will be specific for oxine. Yes. Cucumber cotyledon expansion for cytokinin will come. Okay. So the right answer is what? Draft rice lamina inclination assay. Yes, very good. Draft rice lamina inclination bioassay is used for brassinosteroids. Okay. The crop plant with highest amount of water requirement. General agri question. Which crop requires more amount of water for growing, whether wheat, maize, cotton or sugarcane? What will be the answer? Wheat, maize, cotton. Yes, very good. Sugarcane. Okay, sugarcane will have highest water requirement among this option. Okay. If they give rice, rice will be the right answer if they given rice here. Okay, clear? The pH of 0.1 molar HCl. What will be the pH for 0.1 molar HCl, students? What will the pH for 0.1 molar HCl? Sorry, 10 power minus 1 molar HCl. What will be the pH? What will be the pH? So, if 10 power minus 1 molar, pH means what? pH means negative logarithm of hydrogen ion concentration. This we know. Negative logarithm of hydrogen ion concentration. Okay. H plus concentration here, how much it will be there? Yes, very good, very good. pH will be 1. Okay. It's a simple question and direct question also. Okay. pH will be 1. Yes. Okay. pH is 1. Okay. Which of the following nutrient element remains in ionic form? Ionic form and is main cation establishing plant cell turgor. Okay. For plant cell turgor maintaining, which mineral nutrient is essential? Which mineral nutrient is essential? Yes, very good. Potassium. Potassium is essential for maintaining the plant cell turgor. Yes, very good. Okay. Potassium is important for maintaining the plant cell turgor. Which of the following reagent cannot be, read the question properly, cannot be used for determination of N-terminus of the protein? If you want to determine the N-terminal of protein, different chemicals we will use. Okay. Among them, which we will not use for determination of N-terminal? 2,4-dinitrofluorobenzene. Yes, this is Sanger reagent. This we will use for N-terminal determination. Sanger reagent. Yes, very good. Dancyl chloride we will use, or else dapsyl chloride also we will use. Okay. Phenyl isothiocyanate, phenyl isothiocyanate, Edman degradation. This also we will use. So, hydrazine, this we will not use to determine the N-terminal. This will be used for C-terminal of amino acid determination. C-terminal amino acid determination will be done by hydrazine. Okay. Which of the following sugar is non-reducing in nature? Non-reducing means both the anomeric carbon will be involved in linkage formation. Which among the sugars is non-reducing in nature? Yes, very good. Sucrose, the linkage will be there between alpha and beta 1 to 2 of glucose and fructose. So, both the anomeric carbon of glucose and fructose will be involved in linkage formation. So, sucrose will come. In trahulose, glucose will be linked by alpha beta 1, alpha and alpha 1 to 1. So, both sucrose and trahulose are non-reducing sugar. Yes. So, both glucose and both trahulose and sucrose are non-reducing in nature. Then another general agri question. Different institutes with their location they have given. Different institute with their location. National Institute for Rural Development, where it is there? Yes, this is simple question. Hyderabad it is there. CAU, Central Agriculture University. Yes, it's there in Imphal. Sugarcane Breeding Institute, in last session itself I told SBI is located in Coimbatore. National Institute of Biotic Stress Management, Raipur. Okay. Can anyone tell where the National Institute for Abiotic Stress Management is there? Just comment 3142. Where is there? 3214. 2413. Where is there? Where is there? Abiotic Stress Institute, where it's there? Just let me know where it's there. Yeah, comment. Okay. The absorbance. Then next simple question, calculation question. Just you see this. Okay. The absorbance of 0.1 molar solution of compound X is 0.5. Okay. When measured using a cuvette length of 1 centimeter. Okay. When measured using a cuvette length of 1 centimeter. Another solution is there, that is solution B. Solution A is there and solution B is there. Solution B also having the same component, okay, but with unknown concentration. Concentration is not known in that. It is having an absorbance of 0.15. Okay, if you see in this question, they have given two different absorbance. I am writing here. Absorbance for solution A is what? 0.5. Absorbance for solution B is what? 0.125. If absorbance is 0.5 means concentration of solution A is 0.1 molar. Now they are asking if the same component having absorbance 0.125, what will be the concentration? Okay, what will be the concentration? You just see here, 4 times it's there here, 4 times less. So for this 4 time means 0.025. Okay, directly we can do like this also, or else absorbance 1 by absorbance 2 equal to concentration 1 by concentration 2. You just substitute 0.5 by 0.125 equal to 0.1 by x. Just simplify that, you will get 0.025 molar. Clear? Clear this one, 0.025 molar will come. I hope this is clear for everyone. Okay. Next, which of the following are bioinformatic database? They are asking which of the following is bioinformatics database. They are not using about specific tool. They are asking database, a storage database. What are the databases among this? Entries, yes, this is a storage database. Swissport for protein database, we can use. Gene bank also a protein database, sorry, DNA database. But BLAST is what? It's a tool for alignment, okay, sequence alignment. So this will not come. Okay, so A, C and D, where is coming? Yes, very good. A, C and D will be the right answer. Okay. The crop plant tolerant to available iron in soil is what? Iron tolerance, even iron is there, it will be tolerant and grow. Which crop? Tolerant to available iron in soil is what? Brinjal, sugar beet, citrus, guava. What will be the answer? Brinjal, sugar beet, yes, very good. Sugar beet. Okay, sugar beet is the crop plant that is tolerant to the available iron. Even iron toxicity is there, the sugar beets can grow. Okay. The place or site of oxidative phosphorylation is where? Where the oxidative phosphorylation will happens? Whether in matrix of mitochondria or inner mitochondrial membrane, cytoplasm of cell or outer mitochondrial membrane. Where it will happen? Yes, inner mitochondria. Inner mitochondrial membrane. It will happen in inner mitochondrial membrane. In matrix of mitochondria, TCA cycle happens. This is another important thing. Beta oxidation also can happen. Okay, these are some of the important points you have to remember. Okay. Oxidation of glucose molecule under anaerobic respiration will give how much number of ATPs? Under anaerobic respiration, how much number of ATP will be produced? Yes, very good. Only 2 ATP will be produced under anaerobic respiration. Okay. If aerobic means that will be 30 or 32. Okay, 30 based on the shuttles, which shuttle it will go, based on that whether 30 or 32 ATP will be produced. Clear? The vector that could be used in 2 different system or hosts are called which vectors? This in class itself I told. Different types of vector, binary vector, shuttle vectors, everything I have teased. There itself I have told. The vector that could be used in 2 different systems or 2 different hosts. Yes, very good. Shuttle vectors. Shuttle vectors can be used in different system or different hosts. Okay. Very good. Another important question. VAM, vesicular arbuscular mycorrhizal genus, which plays an important role in phosphorus cycling and its uptake is. Which among is VAM genus? Penicillium, aspergillus. Penicillium will never come. Aspergillus also will never come. Bacillus also will never come. Yes, glomus. Glomus is a VAM genus. Okay. VAM genus. Okay. This is another important point you have to remember. Okay. This VAM, the major function of VAM is what? It will, for phosphorus uptaking, it will help. Okay. The hyphae will help for uptaking the phosphorus. Okay. Statement question there. See, read the statement properly. Nitrogenase is a catalytically efficient and oxygen insensitive enzyme. First statement they are saying this nitrogenase is a catalytically efficient and oxygen insensitive enzyme. No, nitrogenase is what? It's a sluggish enzyme and catalytically inactive or inefficient enzyme. This is wrong, as well as oxygen will be sensitive. So this is wrong. Okay. The reduction of acetylene to ethylene cannot be used as a estimation of nitrogenase activity. No, this we can use. So yes, very good. Both statements are wrong. Okay. Both statement 1 and 2 are incorrect. Okay. So option B will be the right answer. Yes. A cluster of genes under the control of single promoter. A single promoter will be there that will control different genes. What is that? Whether it's a promoter or operator or operon or oncogene or ORF. What will be the answer? A single promoter controlling a set of genes is known as what? Yes, operon, lac operon. We can say in lac operon also 3 structural genes will be controlled by a single promoter. This will be a specific characteristics of prokaryotics. Okay, that will have a operon structures will be there. Okay, Calvin group used the following plant to identify the intermediate in C3 pathway, which they have used, whether they have used Arabidopsis, Chlorella, Garden pea or alfalfa. Yes, Chlorella. They have used Chlorella for identifying the intermediates in C3 pathway. This is simple question only. Okay, simple question only. Another statement question regarding translation transcription. Okay, the cap present at the 5' end of eukaryotic messenger RNA is 7-methyl GTP. Yes, 7-methyl GTP. The 5' cap of eukaryotic mRNA is essential for binding by eukaryotic ribosomes and this also enhance their stability. What will be the answer? Yes, cap structure 5' guanosine will be there, methyl guanosine will be there. Yes, that is correct only. 5' cap structure that will help to stabilize the messenger RNA as well as it will essential for binding of the ribosome. This we have already discussed. Both 1 and 2 will be the right option. Yes, both 1 and 2 will be the right option. Okay. The transfer DNA portion of tDNA plasmid contains gene for, yes, transfer DNA. The tDNA portion, you know, TI vector will be there, tumor inducing vector will be there. Okay, TA plasmid will be there. In that, the particular tDNA will be there, na? What are the genes will be coded by the tDNA? Whether virulence and hormones, virulence and CDKs, opines and virulence, hormones and opines. What will be the answer? Read the question properly. Okay. Particularly, they are asking about the tDNA region, only the tDNA region. We know this tDNA region will go and integrate in the genome of the plant. Okay, plant genome, it will integrated. If it is integrated, what it will happen? Tumor will be formed. Tumor is forming because of what? Hormones, particularly cytokinin and auxin activity. So wherever hormones is coming, only in these two hormones are coming. Okay. Then another most important thing, okay, virulence genes. This virulence gene in class itself I told. Virulence gene function is what? Only for helping for transferring the tDNA into the host. Okay, that will not be integrated in plant genome. Okay, so this will not be present in tDNA region. So wherever virulence is coming, that is also wrong answer. So opine catabolism gene and hormones will be there. Opine catabolism and hormone genes will be there in tDNA region. I hope this is clear. Okay. Another important question from fatty acids. Okay. Plant stanols. Stanol means what? They are hydrogenated. Okay, they are hydrogenated molecules. Okay, that means what? Double bonds will be converted into single bonds. Okay, What they are saying, plant stanols are fully reduced form of plant sterol. Yes, this is true only. It's a fully reduced form. Plant stenols, esterified with long chain fatty acid, can potentially lower the LDL level. This is what bad cholesterol. LDL level in humans by efficiently blocking the uptake of cholesterol. Okay, this is application aspect. Okay, if we consume these stenols, that will act as a competitive molecule. So that will go and bind in the cholesterol binding site. Okay, we know this cholesterol LDLs are what? These are bad cholesterols. Okay, so these stenols, if we go and bind, the accumulation of LDL will be reduced. It will be beneficial for the human. So, this both statements are correct. Both statement 1 and 2 are correct. Okay. An example of model crop in molecular biology. What is the example for model crop? What will be the example for model crop in molecular biology? Rice, cowpea, PGNP, wheat. What will be the answer? For monocots, primarily they will use rice. Yes. For dicot, primarily they will use Arabidopsis. Okay, clear. This is also another important point. Okay, very good. See, match the following simple question only. They have given the different genes and what are the property here it's having. We know cryoneaceae, we have already seen this. Cryoneaceae for insecticide resistance. Okay, cryoneaceae. So, only one thing, cryoneaceae. So, option C will be the right answer. Correct. UIDGA is what? It's a beta glucuronidase. It will act as a marker gene. Correct. BT brinjal, what it will do? BT brinjal for developed for fruit and shoot borer resistance. Yes, shoot and fruit borer resistance. DREB is for what? Drought tolerance. Okay, for drought tolerance, DREB was resistant. Okay, DREB was there. Okay. Another important thing, uh, you just match this. Okay, A, B, C, D. You just let me know the answer. For A, what will be the answer? Okay, A is 3. Like that, you just comment the answers. Okay. Light is the signal that controls stomatal movement in plants. Okay. The stomatal opening is stimulated by which light will stimulate the stomatal opening and closing? Blue light, red light, green light or yellow light? What will be the answer? Yes, very good. Blue light. Okay. Particularly cryptochromes. Cryptochromes are involved in this blue light sensor that will help in opening and closing of stomatas. Okay. The water quality is categorized excellent. General agri question. Excellent for irrigation when the chlorine concentration will be how much chlorine concentration should be there? General agri question. It should be less than 4 or 4 to 7, 7 to 12 or about 12. What will be the answer? 7 to 4 or 7 to 12? Yes, it should be less than 4. Okay, this is general agri question. Okay, just you see once. Okay. Then another important question for cofactor coenzymes. Okay. Urease enzyme. Urease that will catalyze the hydrolysis of urea to carbon dioxide and ammonia. Okay, carbon dioxide and ammonia. Which of the following element is essential part of urease? That means what is the cofactor for urease? Indirectly they are asking what will be the cofactor for urease. This in our discussion class itself I have told what will be the cofactor. Very good. Nickel. Nickel is the cofactor for urease. Okay. Carbonic anhydrase means zinc will come. Carbonic anhydrase means zinc. Alcohol dehydrogenase that will also have zinc as cofactor, but urease will have nickel as a cofactor. Okay. The virus that infect bacteria are bar question, DBT question. DBT bet they have asked this question 2026. The virus that infect bacteria are called cosmids or plasmids, bacteriophages or mycoplasm. Yes, very good. It's the virus that is infecting bacteria is bacteriophages. Okay. Bacteriophages. Okay. Transgenic papaya variety. rainbow is resistant to. Everything is there in our lecture class PPT itself. I have given this. Rainbow is resistant to for transgenic papaya variety. Rainbow is resistant to papaya ring spot virus, aphids, fruit rot, delayed fruit ripening. Yes, it's a ring spot virus, viral resistant one. Okay, rainbow. Okay, rainbow and sunhem. Total 2 varieties are there. So this is very important. Okay, the DNA replication in prokaryotic is of whether it will be semi-conservative, not only in prokaryotic and in eukaryotic also I am saying. Whether it will be semi-conservative or conservative, disruptive or semi-disruptive. What will be the answer? Semi-conservative, conservative, disruptive type? Yes, semi-conservative. This is simple question. Okay, like this question they won't ask, but this is very, very simple. Okay. The reaction, nitrate is there. Okay. This nitrate is converting to nitrite. Nitrate is converting to nitrite. Is catalyzed by which enzyme? The conversion of nitrate to nitrite will be done by which enzyme? Nitrate reductase, nitrite reductase, glutamate synthase or aminotransferase? What will be the answer? Yes, very good. Nitrate reductase. Okay. NR. Okay. This will be present in both root cell as well as in leaf or mesophyll cells. Okay. Assimilation will be happen in both root and shoot cells. Okay. So, nitrate reductase. Okay. NR. Nitrate reductase means NR. NIR means nitrite reductase. That will convert nitrite to ammonia. Okay. This also you have to be remember. Okay. The epigenetic modification which regulates gene expression. Which epigenetic modification will regulate the gene expression? DNA methylation, polyadenylation, splicing of heteronuclear RNA, 5' capping of mRNA. What will be the right answer? DNA methylation or what will be the right answer? Yes. DNA methylation, the epigenetic modification. Okay, particularly DNA methylation will regulate the gene expression, particularly at CPG islands. This they have asked in 2021. Okay, this they have asked in 2021. Okay, if you want to practice topic wise objective question, kindly enroll in our test series. Okay, those who are preparing for ICAR examination 2027, kindly enroll in our courses. It will be highly useful for your preparation. Okay. Next question. The biosynthetic pathway of chlorophyll begins with the compound. That means indirectly they are asking what is the precursor for chlorophyll biosynthesis. Malonic acid, methionine, serine and glutamic acid. What will be the answer? Malonic acid, methionine, serine and glutamic acid. This while class itself I have told you. Okay. The difference between plants and mammals, plants and animals or mammals. I have already told in plants glutamyl tRNA or glutamic acid plus tRNA that will be act as a precursor, but in humans glycine plus succinyl CoA will act as a precursor for heme synthesis. Okay. So what will be the answer? Yes, glutamic acid. Okay. The precursor for heme synthesis is glutamic acid plus tRNA in plants. In animals and mammal, glycine and succinyl CoA will act as a precursor. Okay, that is also another important point. Okay, statement question. In erythrocytes, 7 of the glycolytic reactions operate near equilibrium. That means what? Out of 10 step in glycolysis, 7 is happening at reversible, that is near equilibrium. Yes, this is correct only. The driving force for the glycolysis lies in the reaction which are operating near equilibrium. Whether this will be correct? The regulatory enzymes will not be the regulatory enzyme will never be a reversible process enzyme. This is clear. So, this is not the driving force for glycolysis. If you see hexokinase, phosphofructokinase and pyruvate kinase, these are the 3 irreversible steps in glycolysis. Clear? 3 irreversible steps in glycolysis. That will be the driving force. So, what will be the correct answer? Statement 1 is correct and 2 is incorrect. Clear? This is another important point you have to remember. Okay. The linking number of supercoiling duplex DNA can be equated to is twist and wreath by which of the following expression? Simple question. Okay. Twist means the linking number means what? Twist plus wreath is linking number. This we have already seen. Okay. Linking number means twist plus wreath. Okay. Yes. Twist plus wreath. Yes. Very good. Very good. The alpha helical structure, this is another important question. The alpha helical structure of proteins or result of formation of hydrogen bond between which of the following amino acid in primary structure? Whether it will bind with N and N plus 1, N and N plus 2 or N plus 3 or N plus 4 or N plus 5. What will be the answer? Yes, very good. N and N plus 4, alpha helix. Okay. Then what is this N and N plus 2, N plus 3 and N and N plus 5? This also you have to know. Okay. If N and N plus 5th amino acid join, that will lead to the formation of pi helix. Okay. Pi helix will form. Okay. Or else we can say 4.416 helix. Clear? This will form. If N and N plus 3 is joining means 3 by 10 helix will form. Clear? 3 by 10 helix will form. This means alpha helix will form if N and N plus 4 means. Then N and N plus 2 means 2.2 by 7 ribbon helix will form. Okay, ribbon helix. This is for your understanding only. They may ask these questions also. That's why I am writing it. Okay. So alpha helix means N and N plus 4. Pi helix means N and N plus 5. N and N plus 3 means 3 by 10 helix. N and N plus 2 means 2.27 ribbon helix. Okay, clear? Yes. The plant hormone that activates many defense responses what? What is the plant hormone? Linoleic acid is not a hormone. Cyclic acid not an hormone. So the right answer is jasmonic acid only. Okay, clear? Jasmonic acid for biotic stresses. Okay, particularly insect feeding will be there, na? In that case, that will be happen. Okay, salicylic acid also will come. Okay, these two are very important for viruses and pathogens. Okay, this I already taught in the class itself. Okay. The peptide containing which of the following amino acids are well suited for buffering at physiological pH? If an peptide containing which of the following amino acids are well suited for buffering at physiological pH? Histidine, tryptophan, glycine and cysteine. This also we have already discussed. What will be the answer? Histidine, tryptophan, glycine. Yes, very good. Histidine, TK, because the physiological pK will be around 6.8. TK, this will have a pK of 6.8. So that will have a capability to buffer at the physiological pH. TK, the sequence of amino acid in a protein during translation process is determined by, if a translation is happening, which decides the sequence of amino acid, whether the codons on the DNA, RNA species, tRNA anticodon or mRNA codon. What will decides the amino acid sequence in a protein? Yes, simple question. Yes, mRNA codons. mRNA codons will be there. We know codon means what? It's a triplet of nucleotide. Okay, this will be specific for particular amino acid. Okay. Like this it will happen. So mRNA codons will be there. Okay. Another important question from techniques in biochemistry. Another important question. The fatty acids are converted into which of the following volatile derivatives for separation by gas liquid chromatography? Fatty acids are converted into which of the following volatile derivatives for gas liquid chromatography? Yes, very good. Fatty acid methyl esters. This is very important question. This is repeated question also. Okay. So fatty acid methyl esters will be formed. Okay. Which of the following plant hormone can induce flowering in pineapple? For flowering in pineapple, which of the plant hormone is required? Which of the plant hormone is required for inducing flowering in pineapple? Same question, same here. They have asked two questions. Okay. Yes, very good. Ethylene. Ethylene is inducing flowering in pineapple and flower thinning. We have seen already flower. For flower thinning, we will use naphthalene acetic acid. It's an oxine. Okay, this also you have to remember. Okay. Different mineral nutrition they have given, and they have given the different functions. Okay. See this one, chlorine, magnesium, iron, zinc they have given. We have to match with what are the mineral nutrients that are required for different functions. We know for oxine biosynthesis, which is most important. Zinc is most important for oxine biosynthesis. Yes, zinc is important. So we can easily eliminate remaining all the three options. Okay. Zinc is essential for oxine biosynthesis. Iron, then chlorine. Oxygen evolving species will have this chlorine. Yes, oxygen evolving species will have chlorine. Okay. This will have first. Magnesium. Where it will come? Magnesium. Magnesium will be involved in protein and ATP synthesis because the cofactor for DNA polymerase enzyme requires the magnesium. Correct. Then zinc, then iron. Iron will be involved in synthesis of chlorophyll precursor molecules. Clear? Synthesis of chlorophyll precursor molecule. So A will come option 1, B will come option 2, C 4 and D 1. Clear? D 1. Clear? This will be the right option. Okay. The first commercialized transgenic crop variety is what? First commercialized transgenic crop variety. Bolgard, Flavor Saver, Liberty Link, BT Brinjal. What is the first commercial crop variety? And you have to type, yeah, Flavor Saver, okay. Flavor Saver is variety of which crop? Yes, tomato. Which gene they have targeted there? Which gene and which technology? That also I want. Yes, just comment, yes, Flavor Saver correct, tomato also correct. Which gene they have targeted? By which technique they have targeted? Yes, polygalactonase gene, PG gene, yes. No, not by RNA interference. They have used antisense RNA technology. Antisense RNA technology. Okay, antisense RNA technology. This is very important. Okay, this is very important. That's why I have written here. Flavor Saver tomato variety, first commercialized transgenic crop. They have targeted the polygalactonase gene. So if we reduce the expression of this polygalactonase gene, cell wall loosening will be reduced. So increased shelf life will be there. By which technique they did? Antisense RNA technology. This is another important point. Antisense RNA technology they have done. Okay. Next, the most abundant compound that is transported through sieve tubes. Which component will be transported through sieve tubes most abundantly? What will be the answer? Amino acids, organic acids, proteins or sucrose? Which will be transported more? The most abundant compound that is transported through sieve tubes. Yes, I need the answer. Yes, sucrose. Okay, sucrose will be transported most abundantly in sieve tubes among the compounds. Yes. 35S isotope. 35 labeled sulfur isotope is commonly used in experiment involving which of the following biomolecules? Whether for nucleic acid they will use 35S labeled isotope, or proteins, or carbohydrates, fats and oil. What will be the answer? Yes, nucleic acid. Nucleic acid phosphorus 32 they will use. Whether sulfur is present in nucleic acid? Yes, very good. Proteins. Protein, sulfur containing amino acid will be there. We can use 35 sulfur for that. Okay. A biomolecular complex composed of 4 polypeptides, 2 identical light chain and 2 identical heavy chains is called as what? Whether it's an antibody, antigen, serum, what? Yes, very good. It's an antibody. Okay. Antibody is composed of 2 heavy chains and 2 light chains. Okay. This is very important. Okay. 1433 protein. 1433 proteins are involved in post-translation regulation of which of the following nitrogen metabolism? 1433 proteins. It will regulate which enzyme? Nitrate reductase, nitrogenase or glutamine synthetase. What will be the answer? This we have already discussed in our lecture itself. Yes, nitrate reductase. Okay. NR will be there, nitrate reductase. If it is phosphorylated, this will become inactive. Okay. Here the 1433 proteins will come and bind. Okay. 1433 proteins will come and bind. So, NR protein, nitrate reductase will be regulated by 1433 proteins. Why they have given this name 1433? Based on what? Based on isoelectric focusing and mobility of the protein. Okay, based on this they have given this name as 1433. Okay. In general agri question, in general, the depth of irrigation water needed per irrigation to irrigate 1 meter depth of clay loamy soil. Okay. If clay loamy soil is there, if we want to irrigate for 1 meter depth, okay, at 50% soil moisture availability is what? 50% soil moisture availability should be there. Okay. Generally, the clay loamy soil, okay, generally the clay loamy soil will have a maximum water holding capacity of 160 to 220 mm. Okay, this much of water holding capacity will be there for 1 meter depth. Okay. They are saying 50% of moisture content. So 50% of water holding means half of this one. Okay. So 80 to 110 mm will be there. So most appropriate is what? 80 to 120 mm. Okay, clear. The database used by the entries for bibliographic searches, it's whether it's a PDB. PDB means what? Protein database. Okay, this will never come. TREMBL or Swiss Prot, this also will not come. So it's a what? It's a PubMed protocol. Okay. This is very simple question and direct question also. Database used by the entries for bibliographic searches, PubMed. Yes, very good. Very good, very good. Diffusion of water into protoplasm contributes to which of the component of water potential? This they have asked in 2022 also. Okay, diffusion of water into protoplasm contributes to which component of water potential of plant cell? Solid potential, pressure potential, matrix. Yes, if water is entering in the things in cell, pressure potential will be contributed. Yes. Next question. Which of the following crop genotype exhibit the dimorphic structure of leaf called Kranz anatomy? They are saying which will have a Kranz anatomy. Kranz anatomy will be present in which type of leaves or which plant? Whether it will be present in C3, C4 or CAM. What is the answer? Yes, this is C3, 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 so C4. So, Kranz anatomy will be present primarily in C4 sugar cane. Okay. C4 sugar cane. Yes. The in vitro slow growth culture is a method used for. That means the growth will be very much slow. That will be used for what purpose? Genetic transformation or conservation of plant genetic resource, induction of somoclonal variation or elimination of latent contamination. For what purpose? Yes, it's a preservation method, conservation of plant genetic resource method. Yes, very good. It's a conservation of plant genetic resource method. The symbol psi in T size is stem loop of tRNA denotes which of the following? What's this symbol psi shows? Whether it's an thiouridine, pseudouridine, ribothymidine or inosine. What will be the answer? This we already discussed in our class itself. We have pseudouridine will be there. This we have already discussed. Okay. Majority of the tRNA molecules having the following sequence at 3' end. What the sequence will be there at 3' end of the tRNA molecule? CCA, CGA, GGA, AAA. Yes, CCA will be there. With this A at 2' hydroxy, amino acid will be binded there. Okay, or 3' hydroxy amino acid will be binded there. Majorly 2' hydroxy of the CCA in tRNA molecule, amino acid will be binded there. Okay, clear? Which of the following hormone function as morphogen during embryogenesis? During embryogenesis process, which hormone will act as an morphogen during embryogenesis? Auxin, cytokinin, abscisic acid or ethylene. Abscisic acid will never come, ethylene also will never come. Whether auxin or cytokinin, morphogen during embryogenesis. Yes, auxin is the right answer. Okay, auxin will act as an morphogen during the process of embryogenesis. Okay, another important question from enzymology. Okay, this is one of the like somewhat difficult question. Okay, more advanced one. Okay, we already know this Lineweaver-Berkey plot. We know Lineweaver-Berkey plot means what? 1 by V0 equal to Km by Vmax, Km by Vmax into 1 by S0 plus 1 by Vmax. This we know, this is Lineweaver-Berkey plot equation. This is just a double reciprocal of Michaelis-Menten equation. Vmax by substrate concentration by Km plus substrate. Okay, this is just a double reciprocal. This is Lineweaver-Berkey plot equation. Okay, like this it will come. Okay, this we know. Here what they are asking means Hans-Wolf plot. This is different variant of these plots to determine the kinetic parameters Vmax and Km. Okay, in Hans-V plot, graphing of S by V versus S yield a straight line whose slope and y-intercept. Okay, they are asking in Hans-Wolf plot, what will be the slope and what will be the y-intercept. Okay, you just observe here clearly. Okay, how I am deriving. Okay, we know this is Lineweaver-Berkey plot 1 by V0 equal to Km by Vmax into 1 by S0 plus 1 by Vmax. Okay, in Hans-Wolf plot, we have to multiply both the side by substrate concentration. Okay, so S0 by V0 equal to Km by Vmax into 1 by S0 into S0 plus 1 by Vmax into S0. Like that it will come. Clear? Now we have to simplify. If you simplify all the things, this and this will be cancelled. Correct? This and this will be cancelled. So, Km by Vmax in plus 1 by Vmax into S0 equal to S0 by V0. This is just resembling like this is just a linear equation. If you draw a graph of 1 by S0 against S0, see S0 by V0 against S0, we will get an y-intercept of Km by Vmax and a slope of 1 by Vmax. This is clear. In Hans-Wolf plot, just we have to multiply by substrate concentration. So, if you plot a graph like this, we will get a slope of 1 by Vmax and y-intercept of Km by Vmax. In our question, they are asking first what is the slope value and what is y-intercept. So, 1 by Vmax is the slope, Km by Vmax is the intercept, y-intercept. I hope that is clear for everyone. Okay, this is somewhat advanced question. You have to read. Okay. Nitrate assimilation in plant occurs only in root. Whether it will happens only in root, whether it will happen only in root or both root and mesophyll cells. Yes, it will happens in both root and mesophyll cells. So statement 1 is wrong. Yes. Plant transport the nitrate generated by nitrate reduction immediately. Yes. Plant will transport the nitrate generated by nitrate reduction immediately. Yes, that is true only. It will immediately transfer because the nitrate will may. Yeah, okay, this is clear. This is clear. So plant transport the nitrate generated by nitrate reduction immediately. This is also correct statement. Yes, so statement 1 is incorrect and statement 2 is correct. Lokendra, yes, my interview went very nicely. TK students, any doubt is there? Any doubt is there? Any doubt? With this, we are completing all our previous year question paper from 2025 to, sorry, from 2020 to 2025. Okay. If any doubts are there means kindly let us know. Okay. Thank you. Thank you all.